digestive system in just a moment. When last we looked, we were talking about the beginning of autonomic control of digestion. We selected our food, we put it in our mouth, we chewed it up, we moistened it, lubricated it, broke it into smaller parts, added some initial digestive enzymes, all with voluntary action. Our tongue has moved it to the oropharynx and positioned it for swallowing. That's all voluntary. And in fact, we can hold it there at the back of our throat for quite a while if we choose to do so. But we begin that swallow. We go gulp, and then the body takes over. The first entry point after that, the food ball is partially prepared for digestion. At this point, it would be a kind of a fibrous ball. It would be wet. It would be full of mucus, but it would be something that's prepared to enter the digestive tract. But it's only partly rendered. The esophagus is going to be the beginning of autonomic control. The muscular tube, 10 inches long or so, and the esophagus, when it begins its work, takes that food straight to the stomach. It goes through the diaphragm, a particular opening that grows that way called the esophageal hiatus. The esophageal hiatus then delivers the food into the stomach. If you widen that hiatus, if it gets stretched or the muscle gets fatigued and break down, you get what's called a hiatal hernia. And what that allows is, in the movement of the body, we've talked about the membranes and we talked about the movement of the body. Well, if you open that space above the stomach up, it can actually push through, and you can get a little lobe of the stomach up above that hiatus. And it constricts it kind of into a figure 8 pattern. Food is going to be pushed through the esophagus by waves of muscular contraction that travel down the tube called peristalsis. This is possible when you have those two layers of muscle, longitudinal and circular, laid down in the way it is. We'll talk more about that in just a second. Peristalsis, if you think about this block of food called the bolus, and you contract the circular muscles behind it, you form a little partial wall. Once you set that wall up and contract the longitudinal muscles, you basically pull this point of constriction in this direction. So you move, for this section, you move from here to here. You then contract the circular, contract the longitudinal, and pull it again. So this is actually showing kind of sequential contractions, but it is so well coordinated by the nervous system that it really represents just a ripple of contraction like this that moves more or less continuously, pushing the food in front of it. Now, this is drawn horizontally. So if you draw it vertically, and you know that from the back of the mouth to the top of the stomach, you're basically vertical if you're practicing good feeding habits, sitting up straight, basically with your mouth above your stomach, then you can see that this peristalsis will be very effective. We're going to see this same process, moving material in the body. A primary place where it happens is the urine that is processed in the kidney is moved to the bladder by successive contractions of peristalsis. The esophagus is not well equipped to protect its cells in its lining from acid. And that leads us to acid reflux. The stomach is full of acid, and it's mainly there to clean the food. The mucus that is produced in the stomach is excessive to protect its lining. And if you have anything where the acid of the stomach splashes up past that hiatus into the base of the esophagus, it irritates those cells and they send pain messages. This is called GERD, or gastroesophageal reflux disease. To reflux is to bubble back up. And it's from the acidic, from the fact that the esophagus isn't equipped like the stomach. Swallowing, here's that voluntary phase. You can see that the chewing, the opening and closing of the mouth, the chewing is largely in place, and this bolus of food is being moved to the back of the mouth. You can see that the tongue is very adept, can fill the whole mouth and basically push that food rearwards and downwards into the pharynx. Now, the pharynx is an open tube, but you can see the movement of the soft palate here, up and back, to prevent that food from moving up into the nasal cavity. We see these constrictors working to position the food right above, in the oropharynx, right above the position of the epiglottis and the opening to the glottis and vocal folds. So at this point, as the food moves down, it aids in the pushing of the epiglottis over that opening. And this is where you're initiating the swallowing. This is going to contract the throat, use the tongue, and basically put pressure on the food. The only place for it to go is past the airway into the esophagus. So you'll notice the closed nature or the restricted nature of the epiglottis as that food moves down. Now, also, there's something interesting to notice, and it is actually drawn fairly well here, that the position of the airway is open, but do you notice that the esophagus is wide at the top where it's the pharynx? As you get into the esophagus, it's very narrow. The esophagus only has to bulge when food is moving through it, and the rest of the time it can accordion back in and close. So we're going to see the esophagus spread out as the food moves down. The esophageal phase and the initiation of peristalsis as the food moves down. Notice this constriction is moving with it, keeping it going. And also notice that with the food right here, the epiglottis has already popped open, opening the airway. Shown here, a bolus of food over the epiglottis, the esophagus largely closed, the airway open, the pharyngeal phase. When you enter that, you initiate that swallow and reflex takes over. Bam, the epiglottis closes the airway. The food moves quickly beyond it. And you can notice here the airway immediately pops open. You'll notice here the esophagus is bulging around the bolus, and that is going to assist the contraction in keeping this bolus moving. It's going to be continuous through the esophageal phase until it basically dumps the food right into the stomach. The bolus enters the stomach through the cardiac sphincter here in the diaphragm, and we're ready to talk about this large, kind of the largest diameter bag in the digestive tract. It receives the food from the outside. It enters as a bolus, and one of its jobs is dilution. So the fluids that are being added are going to change it from that kind of fibrous mass, a more solid bolus, into a flowing gastric stream of liquid. We're going to call that mixture chyme. Technically, it's a slurry because although it does contain lots of dissolved molecules that are dissolved in the water solvent, it also contains lots of particles, but it's very, very liquid at that point. The cardiac sphincter is where the food enters the stomach. The stomach is going to continue mechanical digestion. You can see here's the longitudinal layer here's the circular layer, and in the stomach, here's a third layer called the oblique layer. It's oblique because it's not parallel to either one of the other layers. It crosses them both at an angle. And with the three layers contracting in sequence, it produces an actual churning, an actual kind of thrashing of the stomach, because there are so many things you're adding to it, and you want to be it thoroughly mixed. You want those solid particles to break apart and be completely coated with the stomach acid, with enzymes that are being added here and other additions. Mucus as lubricant is a good example. Gastric juice is what is the kind of generic term, and the chemical digestion is greatly amplified by what's added here. The acid does have a lowering of the pH effect and a killing of microorganisms. Well, we also add a general group of enzymes called pepsins. Pepsins break peptide bonds and therefore begin protein digestion here in the stomach. The wall of the stomach is complex. You can see the outer visceral peritoneum and the connective tissue layer that's going to embed and basically secure the blood vessels, lymph, and nerve elements that are present in this organ. Here is longitudinal and circular, and here is the oblique layer, more connective tissue in the submucosa, with ample blood vessel and lymph populations shown here. Now, this is really making sense. We're going to actually maintain a big population of living cells all through the walls of the digestive tract. But think about when we get to the small intestine, we're also going to pick up those nutrients and bring them across and put them in blood vessels. So we're going to have a solid connective tissue layer. Notice that the mucosa is greatly expanded here. This expansion of the mucosa is to accommodate these deep pits. These deep pits are lined with populations of glandular cells that will perform the functions of the stomach. So here is the closed inner surface of the stomach facing the lumen. This is where the food is being mixed. And what's coming out of these pits is the purple cells, notice, that line the neck right to the opening of the lumen of the stomach, are the mucous neck cells. They're pumping out mucus like crazy. That mucus comes out, and the first thing it does is it protects these surface epithelial cells of the stomach from the acid conditions. Now, it is a kind of a losing battle, because the bolus / chyme is scraping across here and removing that, and so more is being exuded all the time. This is a good covering for the lining of the stomach. Chief cells, shown in pink, are abundant, and they're producing pepsinogen, a precursor to the pepsins that we mentioned, an enzyme of protein digestion. The green cells are parietal cells. Notice there's not many of them, but they are producing hydrochloric acid. They're going to use the hydrogen ion that is generated by the bicarbonate system. Carbonic acid dissociates into hydrogen ion and bicarbonate. Well, they're going to grab that hydrogen ion and shove it into the lumen. And to keep it in solution, you're going to have to accompany it with a negative ion that's makes a strong acid. And you're going to take chloride from the sodium chloride that we eat to make the hydrogen chloride or hydrochloric acid mix that will make this very, very acid. These are parietal cells. Finally, gastrin are shown being produced by the cells shown in yellow, the endocrine cells, and those endocrine cells in the mucosa. Gastrin is a regulatory hormone. So the presence of gastrin, or the passage of gastrin out of a region of the digestive tract, will have feedback implications through the plexus of nerves that communicate between different sections, and will have effects on everything from secretion to the activity of the muscular layer as food passes through. When you think about a meal, let's say I sit down at noon and I'm going to have a lunch, and I eat from 12:15 to, let's say, 12:40. During that time, I'm chewing and swallowing, but there's definitely a beginning and there's an end to my meal. So you have to activate the digestive tract in preparation for the beginning of feeding. You then have to keep it active, secreting and massaging, contracting through that meal. But then when the last of the food moves through, you have to shut it down. Refill all of those glandular pockets and put it at rest until the next meal. The secretion of hydrochloric acid is shown here by the parietal cell. It comes from this CO2 plus H2O going to carbonic acid. Remember, this was happening in the red blood cells as well, producing bicarbonate and hydrogen ion. Well, the bicarbonate is pumped out into the bloodstream. This is called the alkaline tide because this is a basic element. And in order to balance the charge, a co-transport mechanism moves chloride into this parietal cell. So you've got hydrogen and chloride in here, and you're basically going to pump those out using a specific transport mechanism. Notice to move that hydrogen ion, it's active transport. You have to use ATP. There's no way this is diffusion. You're trying to pump all the hydrogen you have across into that lumen to make it a very acid condition. pH 2, I've even seen some references that put stomach acid at pH 1. So you can see that there's an active movement and exchange to balance the charge and get the chloride from the salt we eat from the bloodstream into the parietal cell, and then to transport it into the lumen of the stomach. Parietal cells also produce a thing called intrinsic factor. It's involved in the vitamin B12 pathway. So intrinsic factor from the stomach is part of the story we tell to vitamin B12, and a shortage of vitamin B12 produces a kind of anemia called pernicious anemia. It is the digestive tract that contributes to that blood condition. When we talk about gastric activity, I'm thinking of sitting here. It's been now 5 hours, 6 hours since my breakfast. My stomach is sitting at rest. If I'm healthy, the hydrochloric acid and the pepsin and the intrinsic factor and the mucus are all ready to go. But the stomach's not doing much. There is a central nervous system contribution to gastric activity that occurs now. The stomach may be sending me messages that it is empty, that are related to what we call hunger, and hunger will lead to food-seeking behaviors. Now, often we're not really hungry. We run by the clock. Noon is about when I eat my lunch. And so we go through food-seeking behaviors. Now, we call this the cephalic phase because it involves our sensory system. So I notice this especially when I go home and a meal is being prepared. Or when I walk into a restaurant, you have the sight of food, you have the hunger pangs, you have the smell of food. The anticipation is something that's being propagated by your central nervous system. And it's sending messages through the vagus nerve, N10, down to this plexus of nerves called the submucosal plexus. So it is that second layer that's between the, for most of us, in this case, the the oblique muscle layer in the stomach and the submucosa. And it's going to send messages to mucus cells, chief cells, parietal cells, and G cells, and all of these secretions are going to begin. So this is kind of a building on Pavlov's experiment. Pavlov was the Russian scientist that showed that you could induce dogs to salivate by presenting the clues that food was on the way. And then he used that to condition. So those cues for food were associated with something like rattling the keys. After a period of condition, this cephalic phase would cause the dogs to salivate if you just rattled your keys. So definitely a nervous system and a learning system precedes the arrival of the food. When the food starts to arrive, we call that the gastric stage. And what it generally does is it contributes weight and volume to the contents of the stomach. That means the stomach stretches out as the food drops in. That activates two things: stretch receptors as the stomach wall stretches, and chemoreceptors as the chemicals arriving in the food are detected. It also dilutes the pH of the acid that's in the stomach. So the the nervous stimulation from these two receptors are moving over to the submucosal and mesenteric plexus, the local nerve networks. Notice this is a local effect. This is not an additional message from the brain or spinal cord or any of the ganglia. It actually is moving from one stomach region to another. A good example of local regulation by region. And these amplify the previous signals. So mucus, pepsinogen, and HCl, all gastrin, all begin to dump in high waves. Now, what gastrin entering the small intestine does is it feeds back a signal, which is an inhibitory signal. By the way, I didn't mention, but these signals, in addition to the secretions, stimulate the muscle layers, all three of them, to begin their sequential contractions, that churning and mixing. So we've got a thorough secretion and mixing picture here, and a dilution of bolus to chyme as it moves toward the exit point of the stomach. Finally, as that food moves through, there's a kind of a continuous movement, continuous movement, continuous movement. And as long as food continues to arrive, this gastric phase is reinforced. So gastrin is, in its way, secondarily amplifying the secretions all the way through this gastric phase when food is arriving. However, the intestinal phase, when the last of that food passes through, the presence of lipids and carbohydrates here in the small intestine, the decreased pH from as you feed, the pH goes higher and higher and higher as that hydrogen chloride is carried away. And there's a feedback message from the small intestine. Again, this is still local because it's within the same system, but via the bloodstream, it comes back and it begins to inhibit chief cells, parietal cells, and the peristalsis of the muscular system. So we see there's a way of inhibiting gastric activity as the food passes out of it and leaves it behind. The arrival of chyme in the small intestine also does another important thing. It stimulates this first region, let's say 6 to 7 inches of the small intestine. This is called the duodenum. And the duodenum has ducts from the liver, a common duct that results from the joining of the duct from the gallbladder, which stores bile from the liver, and a duct from the pancreas, which stores all kinds of digestive enzymes. Let's just call it pancreatic juice. Many, many specific lytic enzymes that will contribute to the chemical digestion of the chyme, but also that insulin that we mentioned earlier. So the intestinal phase continues throughout the feeding cycle. In general, one section of the intestines will receive the food, will secrete, will absorb, will lubricate. As the food moves past that section, it will be feeding back to the previous section, shutting it down, and sending nerve impulses to the next section, starting it up, so that the activity of the digestive tract kind of follows the food back and forth as it takes its basically downward track through the small intestine. Ulcers are a condition, used to be thought of as a stomach condition, and we used to say hurry, worry, and curry could cause ulcers. If you're stressed from trying to do too much, being overworked, if you're worried about something, so you're emotionally upset on a constant basis, or if you eat a lot of spicy food, they thought it caused ulcers. Now, that was not a very good diagnosis, and the various treatments for that were not very successful. The pH in the stomach destroys most of the microorganisms that arrive there, but we find that ulcers are correlated in a high degree with a particular bacterial infection, Helicobacter pylori. And this produces an enzyme called urease. Now, think about urease. It's an ACE. So what do you suppose it breaks down? It breaks down urea. When you break down urea, what do you get? You get ammonia. So if you get a cell lodged on the wall of the stomach and it starts breaking down urea to form ammonia, it neutralizes that microenvironment. And so it produces a little more neutral zone where the bacteria can establish. The establishment irritates the wall of the bacteria and produces the pain and discomfort that are characteristic of ulcers. Now, it's not a one-to-one correlation. A lot of comparisons of this slide with what we've been hearing about COVID-19. Wouldn't it be nice if you either had it or you didn't? Wouldn't it be nice if you tested positive and you had symptoms? I mean, right now we have such a mixture of asymptomatic people that are testing positive. They can even communicate the disease. We see the same kind of sort of partial effect, disease effect, from Helicobacter. 10% to 20% actually develop ulcers. If you start testing them, you find a lot more people who have it than complain about ulcers. Individuals with stomach cancer, however, 90% are infected with this. Now, that's too large a number to dismiss. So, although that does not mean that the presence of the bacteria will give you stomach cancer, it is a positive influence on the development of this particular kind of cancer, a mutation of a cell within the stomach. Stomach cancer is a very serious and deadly cancer. Mechanical digestion in the stomach from churning, from the muscular contractions and the acid environment. Chemical digestion from the pepsin, and the food leaves as a slurry called chyme. It moves through the lower opening into the small intestine, an area called the pyloric sphincter. Now notice we've moved through hiatus and sphincter areas, meaning we can contract the lower part of the stomach to restrict the flow, but there's no wall, there's no flap, there's no valve. It's basically an open tract all the way down. Small intestine is an extended small tube, and that small tube increases the surface area. It consists of three major areas. Up here, from the stomach, these first few inches is called the duodenum, and this is an area of major additions. This is where the bile comes in. This is where the pancreatic juice comes in, and this is literally dozens of chemical additions. Digestive enzyme, chemical digestion depends strongly on the materials added here. Then the jejunum, an extended small intestinal region, small diameter tube, winds back and forth, suspended by its mesenteries. By the way, you can kind of see the sort of fan-shaped nature, as you'll have a fold And the blood vessels supported in that serous membrane to basically populate the capillaries in these first few inches of walls, and then another fold that goes the other way out of sight and then comes back out this way and curves around. You can see how this serous membrane is acting to support the weight and provide blood and nerve services at the same time. The jejunum is where most of the absorption occurs. Finally, down at the bottom end, you have the ileum, the final stretch of small intestine. Some late digestion and processing does occur here, but the ileum joins to the large intestine through this ileocecal junction. This first lobe of the small intestine of the large intestine is called the cecum. This little finger-like projection is the appendix, which is just a remnant tissue, largely non-functional in our body, and also one that gives us trouble when it becomes infected. So let's look at the duodenum, ileum, the duodenum, jejunum, and ileum individually. The small intestine is built for absorption, and so we're going to look for increased surface area. In cells, we talked about folds and membranes. We talked about finger-like projections. We're going to see that at several levels because most of the absorption occurs in the small intestine. In fact, it's so decorated here at the top. The brush border is shown here, these finger-like projections. Now, these would be villi and microvilli. These are membrane tubes that are covered with a thin membrane to facilitate that absorption, and also supported from within. Also, we're going to have capillaries inside every one. So the tube itself is convoluted like this. Those folds and ridges you can see with the naked eye are called plica, and you can kind of see them here in magnification. But when you blow it up further, you'll notice that even these bumps, the plica, are covered with smaller extensions, the villi and microvilli of the intestinal lining. Now, it looks like this. Here you can see a lymph vessel, and you can see an artery vein system which extend to capillaries. Lacteals is what we call the lymph capillaries, shown yellow here. And red and blue is basically artery and vein flow through this individual villus. Notice that single cell layer. Notice the goblet cells and their mucus, because it's important. I mean, think about this food, these solid materials moving through this that looks like a deep pile carpet. You don't have them lubricated, then that muscular contraction from these two muscle layers, the circular and the longitudinal, is not going to keep them moving. Okay, all in all, this is our absorption region. The duodenum is that first region, and by the way, I'll point out, the stomach, where the pointer is now, is inside the peritoneum. But right here it exits into the retroperitoneal space, and the duodenum is hooked through the common bile and pancreatic duct in the retroperitoneal space. And then right here, it changes to the jejunum as it ducts back in to the peritoneum and remains there. These particular illustrations show that jejunum, actually by this time, there have been a lot of monomers generated. And if they are simple, straightforward monomers, things like glucose, things like amino acids, things like other monosaccharides, they literally it's like they get sucked into the capillaries. Now, oil, fats, and waxes have been processed by the bile for only a short time from the addition from the common duct up here. But they're beginning to be emulsified. They're going to be processed further and further as they move through. And they're going to be a sequence of names for these little droplets. They're not really being chemically processed down to acetyl groups or fatty acids. They're just being suspended as tinier and tinier droplets, eventually called chylomicrons. I'll show you that in a second. So along this length, those fats, oils, and waxes are being absorbed, are moving into the intercellular spaces, but they're picked up by the lymph system and basically transported out of the peritoneum up to the thorax, where they're dumped into the blood. The small intestine, this slide says the first 12 finger breaths, I don't know how much that is the duodenum. It receives the acidic chyme, and normally, by the way, chyme is spelled with a Y, and bicarbonate buffers are added. Notice that particularly. We've just acidified the material in the stomach down to pH, well, depending on how much we ate and drank, but we took it from pH 1 or 2 up to 3 or 4 as it exits. We're going to then neutralize it right back to the same pH as the blood plasma, 7.4. And we're going to use bicarbonate buffers, that alkaline tide we spoke about, to do that neutralizing. We also get the bile and the pancreatic juice added, and we are going to do this in the duodenum. The greater omentum, this is kind of showing a little more drawing. The omentum normally hangs down here, it kind of covers the abdominal organs, sort of like an apron. Pull it up and back. Here's the transverse colon, and here is the transverse mesocolon. The duodenum is this region of the small intestine right after the stomach. The jejunum's about 8 feet of our intestine, and look at the plicae. Look how folded they are, and are microscopically loaded with villi and microvilli to increase the surface area to a very great degree. This is where most of the chemical digestion and absorption occurs. So when we say digestion, it means we're still taking those saccharides and disaccharides and breaking them down into monomers. We're still taking dipeptides and tripeptides and breaking them into amino acids. But then we're putting them across the wall into the space around the capillaries where they're absorbed and carried away. The ileum is a significant portion of the intestine, and you can kind of see the importance of propulsion here as they're drawn here. Look at this. This comes down and then goes straight back up, loops down and comes back up, down and up. So we're having to repel this liquid chyme, and it is very watery at this point. And it is the muscular contractions that are squeezing it and squirting it along this pathway, eventually to this junction right here. We do reabsorb bile salts. Bile salts are taken back out of the digestive stream where they're basically treated as waste. And the processing of bile is something that happens in several places in the body. It produces a substance called urochrome, or urinary color. It contributes a yellow color to urine. It contributes eventually a yellowing and browning agent to feces, and it is present in all of our eliminations. This is where we also absorb vitamin B12 in the ileum of specialized regions. And this is a place where we start to pick up some additional malt or lymphoid tissue. Now, let's make it clear. What harbors the largest population of microorganisms are these large intestines right here. This is where our gut flora lives, from the rectum all the way back to the junction with the small intestine. Up here in the stomach, we just tried to kill all the microorganisms. But from the large intestine, and this cecum and this valve is really more of a sphincter, so bacteria actually can go backwards into this small intestine. So we see more lymphoid tissue called Peyer's patches as a means of controlling those microbial populations in the small intestine. Now, when we talk about nutrition, And when we talk about absorbing nutrients, I want to make one point very clear. Digestion is specific. Digestion is specific. Like the chemical reactions in the body, there are enzymes to do a specific job. When we look later at metabolism in AMP2, we're going to notice that the so-called Krebs cycle has eight reactions, and every one of them has its own enzyme to make sure that this intermediate goes to the next step in the cycle. The same thing applies to the delivery of our nutrients. Our food consists of three main nutrients. Now, you'll notice this is three of our four big four, and this is the one that's missing is nucleic acid, and there's a simple reason for that. We register our food value based on calories, how much heat is released when we burn it. Now, nucleic acid, it turns out, and the nucleotides that make it up contain lots of energy. But compared to carbohydrate, lipid, and protein, there's just not much of it in each cell. And so with respect to burning a gram of tissue to figure out its contribution, nucleic acid, we don't actually have that much nucleic acid, even though there's a complete set of blueprints in the nucleus of every cell. So food consists largely carbohydrate, and here's starches and sugars. We have to break them to monosaccharides. Lipids are going to be broken down into fatty acids. These are those long carbon hydrogen chains. And monoglycerides. That means something like a glycerin with fatty acid chains on it, something called a triglyceride. Proteins are broken down into amino acids. Amino acids are the nutrient. Now, this is a very busy figure. I'm not sure this figure number corresponds to the one in your edition, but it really deserves a good hard look, because this is the chemistry of digestion. And in addition to the physiology, it shows you the anatomy as well. Let's take this first purple column on the left. Carbohydrates receive amylase in the mouth. Nothing except transport in the esophagus. And in the stomach, we're already talking about disaccharides and trisaccharides as that starch is just falling apart. In the small intestine, we get another enzyme called pancreatic alpha amylase, which is very efficient in breaking down these polysaccharide chunks. So we're still at disaccharides and trisaccharides, some monosaccharides, but at the brush border of the intestinal mucosa right here, we have specific enzymes like lactase, maltase, and sucrase that break them down to their components. They break them down to the monosaccharides that make them up. Largely glucose, fructose, galactose, other single monosaccharides. Now, we can move those by facilitated diffusion and by co-transport for the various carbohydrate, the monosaccharides that we see here. We're going to have specific carriers in the brush border. So there's going to be a specific glucose transport carrier. And because it's always high concentration, arriving here with the food stream, and lower concentration here, because we're going to take these monosaccharides as they accumulate here in the cell, and facilitated diffusion is going to move them right into the spaces next to the capillaries where they're absorbed and transported away by the flowing blood. That works very well because glucose and other monosaccharides are hydrophilic. They move well as a dissolved carbon molecule. I'm going to skip over the central column just now and go to proteins. And I'm going to proteins first because it's hydrophilic in its amino acid form. It's going to basically take the same pathway as the carbohydrate. So here, we don't really get any significant protein digestion in the mouth or the esophagus, but in the stomach, pepsin arrives, and polypeptides are broken down into shorter peptide strings. When we get to the small intestine, pancreatic juice has a number of specific protein digesters: trypsin, chymotrypsin, elastase, carboxypeptidase. Now, why so many? Well, you remember we have 20 amino acids. The peptide bond is consistent between amino acids in making that string. But the R group affects how you can attack that peptide bond. So if it's between a tyrosine and an aspartic acid, you may have to have a specific enzyme to break that bond. And so there are more. But once you get to these short peptides and amino acids, you have dipeptidases, or the dipeptides, right at the brush border, right on the intestinal mucosa, and you end up with facilitated diffusion and co-transport, depending on the entity and the carrier, to move these into the cells, and the same passive processes. Notice these are both diffusion processes, no energy required, to move amino acids out into these spaces, into the bloodstream, and away with the enriched blood. So coming out of the small intestine, you're going to have a rich array of monosaccharides and of amino acids. Now let's go down the lipid pathway. Lipase is part of the lingual enzyme secretion. In the esophagus and in the stomach, not much happens. Fats, oils, and waxes are there separating from the water mix of the bolus and the chyme. But once we get into the small intestine, bile salts and lipases in the pancreatic juice begin attacking. And pretty soon, monoglycerides and fatty acids are rendered. Now, the bubbles that they occur in, they're still hydrophobic. So they're occurring in bubbles, but because of the bile, they're getting smaller and smaller as you move into the small intestine. We call this individual little droplet a micelle. We're going to process them further. Diffusion is going to move this. Remember, the membrane's hydrophobic, just like the micelle. And so it's basically going to just drop right through that membrane by diffusion. Monoglycerides and fatty acids, triglycerides, are formed. This would be a good example of glycerol with 3 fatty acid chains, and processed into even further smaller drops called chylomicrons. Now these are, as I said, hydrophobic still. So it's going to take exocytosis, tiny vesicles, to expel them from the cell. But an interesting thing happens right there in that villus, or that microvillus, where there's a artery and capillary and vein network and a lymph vessel. The chylomicrons enter the lymph. They are a little more soluble because of the triglycerides. The glycerol end is a hydrophilic end, and the fatty acid end is a hydrophobic end, but it does move somewhat through water. So it's going to enter as a chylomicron in the lymph, and instead of going through the cardiovascular system, it's going to take that lymph network, ascending up through the abdominal cavity. Well, remember, the entire abdominal cavity is served by the left lymphatic system. And so it's going to basically ascend up here, where the major lymph ducts dump into the vein system in the subclavian region. So in addition to returning all that excess water in the form of lymph, it's carrying our lipid nutritive component, and this is where it enters the blood, not the walls of the small intestine, but up in this vein or lymph vessel to vein connection. Digestive systems adapt to the different diets. Herbivores, like this squirrel, eat vegetable matter. And like I say, plant cells are all covered with cell walls. In order to get nutrition out, you can't digest the cellulose. You got to chew down on and treat with harsh enough chemicals. that you rupture the cell and let the nutrients out of the living cell. The cellulose is just going to go out the other end. So that takes more time, more mechanical disruption, more, in some cases, more chewing. It's going to take more time for chemical digestion. And so you see for a squirrel, given the overall body size of a squirrel, a long, convoluted digestive tract. I said more chewing. If you've ever watched a herd of cattle, they will feed out of the trough or off the ground as long as fresh food is available. But after that food is all gone, sometimes you'll see a cow standing, or you'll see it basically down on its belly, and it will all of a sudden just start chewing. And it will chew and chew and chew, and then swallow and stop chewing. Do that throughout the day. That's a process where the cow actually takes the partially mechanically disrupted grass or hay or grain and brings it back up into its mouth by regurgitation, chews it again to release more nutrients, and swallows. It's a method of getting more nutrition out of the nutritive material. Now over here we see a carnivore. Carnivores are meat eaters. So what they're eating is a much closer tissue correspondence to their own tissues. So here you're having to convert plant nutrients into animal tissue. Here you're feeding on animal tissue. And so the presence of the monomers and polymers that you need is in higher concentration. So you're going to have to work to get it. However, you can digest it with a much shorter digestive tract. Notice that this wolf shows a reduced length of digestive intestine compared to the squirrel. There's also another feature here, and that is this cecum. It's extended in the squirrel because it's very active. If you eat additional plant material, more than you need, you can actually fill the cecum up with stored material, stored but partially undigested food stream, and then basically press it back out into the digestive tract for additional nutrients at a later time. The cecum in the squirrel is very well developed and is in general in herbivores. The cecum is better developed in wolves because wolves still are feeding on an as I can catch it basis. What happens if you're feeding on a herd animal like caribou and then the caribou move off? If they move off and you're denning, you may lose contact with them. And so storing in this cecum, in this esophagus, not esophagus, I'm sorry, appendix is something that was in the past of human beings, although we do notice now ours is greatly atrophied. The large intestine is a much larger diameter tube. It appears in lobes, but it does still have a slightly kind of folded appearance in the inner membrane. Here is the presence of a very strongly reinforced valve, the ileocecal valve. It still is a sphincter. It isn't an absolute barrier, but it regulates the movement of material from the small intestine into the large intestine. And it must move from this point through the ascending colon, the transverse colon, and the descending colon, ending up in the anterior position on the lower left abdomen. Notice the sigmoid is drawn just as a curve here. It really is more of a snaky thing, positioning the flow of material over the rectum, the large muscular sac, and the anus, which guards the opening. The large intestine is going to do an important thing. We have taken in and used a lot of water. We've taken our food material and added liters of water in the digestion. And as we extract the nutrients, we don't need as much water in order to have it move smoothly. But we also are moving, we are diluting the presence of waste materials, it's becoming much less than up there in the stomach at the top end of the tract. So through the small intestine, but then finally in the large intestine, we reabsorb that water, step of water conservation. If we ate a meal and we threw away 2 liters of water every time, we would have to be drinking much more water. But this is not always balanced and healthy. We want our feces to be a semi-paste. So we call it solid waste, and the actual solid particles are very small in most feces. However, we do want it to be a pliable paste, like toothpaste, something that if you squeeze with muscles, you can cause it to come out the other end. If you reabsorb too much water, it leads to a compaction of these particles into what's called constipation. The lobes of feces can meld together based on pressure and become too large to easily get out the anus. If you absorb too little, you lead to diarrhea. Diarrhea is watery stool, and what you're actually seeing is more the consistency of chyme. Chyme as it's moving from the top of the small intestine, certainly the beginning of the jejunum, all the way down through the large intestine. It's a watery material that's flowing with suspended particles in it, a slurry. So if you shut down this reabsorption in the large intestine based on some kind of gut infection, what comes out, that watery stool, is more like the chyme. We do see some odd things in the large intestine. There's a gut flora. We used to say there are dozens of species of bacteria, and that was based on a very, very restricted definition of what constitutes proof that that bacteria lives in your gut. Now that we have DNA sequencing, it's much easier to just take a fecal sample and pull out and isolate individual cells and let them reproduce and do DNA analysis. And now we know that it's certainly hundreds of species in a healthy gut. It's the whole idea behind probiotics, isn't it? That you're adding some kind of bacteria that will reside in your digestive tract and in some way improve the condition of your tract. One of the things that happens in the large intestine is there's a particular bacterium that produces vitamin K. Now, vitamin K is part of the blood clotting sequence from the forming of fibrinogen and then during an injury, the formation of fibrin fibers. This occurs in the liver, but vitamin K is supplemented by this bacterial production. Now, if you think back, there was a term symbiosis when 2 species live together. And this would be an example of mutualism. Mutualism is one where the benefit to the bacteria is a terrific place to live. It's warm, it's dark, it's wet, its conditions for growth are ideal, and it has a constant stream of food going through it. The advantage to the human being is that it gets vitamin K, and it's notable to say this vitamin isn't involved in any diagnosis of disease that we know about. So it does not hurt us, but it gives us vitamin K. So when we look at the balance of water movement in the digestive tract, we come up with a picture that looks like this. This particular figure shows us water addition and water extraction or reclamation on a daily basis for a typical average person. So I notice we're getting late in the period, so I'm going to end this show. Today's is